the construction of your world model and why that's really important. Then we will look at the effects of psychedelics specifically on the construction of that world model. So how do psychedelics affect the structure and dynamics of your world, basically, of your personal reality? And then towards the end, perhaps opening it more up to discussion at that point, think specifically about DMT, which has this extremely profound effect on the world model. So that's the idea. I don't know how. I'm happy for people to raise their virtual hands and ask questions throughout this. I don't know how that works, but
we can, as the facilitators, we can be monitoring the chat and the hand raising stuff, and we'll just filter that to you, so you don't have to worry about it.
Yeah. Okay. So please, if I say something daft or something incomprehensible, then do stop me and do ask. Otherwise, I'll just kind of rattle on. So I expect to talk for an hour or so, and then, but who knows? Okay, so let's start. So kind of I guess the fundamental axiom for anybody, in my opinion, for anybody that is studying psychedelic drugs, is an appreciation of the fact that as a conscious organism, you are immersed within a world. This is your own personal, subjective world, often called your phenomenal world. And whenever you are awake, or even when you're dreaming, you are always immersed within a world, whether it's normal kind of sober waking life, whether you are under the influence of a psychedelic drug or some other kind of brain stimulating or perturbing process or molecule, you're always immersed within a world, and the nature of that world can change depending on conditions, but you're always within the world. And so understanding psychedelics is about understanding, first of all, what is that world built from? What's the structure of the world? What's it for? Where did it come from? And then to understand how psychedelics actually affect the structure of that world. So that's kind of fundamental stuff. Second kind of axiom based upon the first one is that this phenomenal world, this personal subjective world, is always built from information that is generated by your brain. That's always the case. This is demonstrable that that's the case. Now that doesn't mean that we're kind of discounting the role of consciousness. Indeed, consciousness is extremely mysterious in some senses, in that for some reason, this information that your brain generates that represents the structure of your phenomenal world, for some reason that we don't really understand, has a subjective quality to it. You know, the brain generates this information, and for some reason, this highly complex pattern of information is experienced from your subjective perspective as your phenomenal world. That's still something of a mystery, but we know from decades of neuroscience research now that if you perturb the brain, you perturb the informational structure of the world. You know, if you damage certain parts of the brain, for example, you can remove certain features of the world. You know, it's quite clearly demonstrable that the informational structure, the content and structure of your phenomenal world is built by a brain using information. So this phenomenal world has a purpose, and you know, the brain is a product of evolution, and the purpose of this phenomenal world is as a model of the environment. Your brain evolved to do many things, but one of the primary functions of your brain is to construct a model of the environment that allows you to navigate the environment. You know, whatever it is out there, we can never really access the external world in itself, the noumenon in Kantian parlance. We only have access to the phenomenon. We only have access to the phenomenal world, which is this model of the environment, a kind of simulational space that we exist within. And this is the model that your brain constructs from information. So let's get down to the nitty gritty a little bit. So when we say the brain is constructing a model of the environment, we're really talking about this outer layer of the brain, the cortex, the gray matter, which is responsible, which is the kind of the world building machinery that's responsible for construction of this model of the environment. Now, of course, if we go in and look at the cortex deeply, at the sort of microscopic level, we will see that it's actually constructed as this highly complex network of molecules of neurons that are interconnected. And the neuron is, of course, as you should all know, the fundamental information generating unit of the brain, using action potentials, basically. So the neuron receives information from other neurons via its dendritic tree, processes this information at the cell body, and then transmits the information via its axon to other neurons using the synaptic connections. But this is all basic neurobiology. So in particular, we're going to be looking at a structure, a very important structure in the cortex, which is these cylindrical structures, which are built from large numbers of these neurons. So each of these structures, which are called cortical columns, is constructed as a network of neurons. And the cortical column, and there are many, millions and millions of these cortical columns packed together kind of sideways in the cortex, and they are heavily interconnected. And the cortical column is thought to be the fundamental information generating unit of your world model. So each cortical column is often called the fundamental unit of functional segregation in the cortex. So there's no smaller feature of the world can be constructed than can be constructed by a single cortical column. So cortical columns, they can be kind of activated or inactivated in varying levels of activation, and they are, as I said, interconnected, so they can speak to each other. They can pass information from one cortical column to another and affect the activity of other cortical columns. So if you look at cortical columns from a kind of bird's eye perspective, or from the side, in the lower half of this diagram, you'll see that the cortical column has a number of layers. There are actually 6 defined layers. Layer 1 closest to the surface of the cortex, and layer 6 being the deepest layer. But we're mostly going to be looking at them kind of from a bird's eye perspective. So this top layer of circles with a white outer ring, these are cortical columns, and we're going to be using this representation of cortical columns basically throughout this talk. So if we were to take a very small patch of cortex, we would see these cortical columns kind of packed together, forming this kind of mosaic structure. And the way that we will represent the activity of these cortical columns is in the gray state, we'll say, is kind of an off state, whereas a yellow state would be an on state. So these cortical columns, they can kind of activate and deactivate. They can switch on or they can switch off. And when because each cortical column will either exist in an active or an inactive state, it forms this pattern of activation. It forms this pattern of information. And it's this pattern of cortical column activation that represents the informational structure of your world. So from moment to moment, what the cortex is doing is moving through patterns of activation, individual states, if you like, of the cortex. And each state of the cortex, each pattern of cortical column activation actually contains and represents the entire informational structure of your world at that point in time, which is quite a remarkable thing. So let's have a look at this a little bit more deeply. So as I said before, the cortical columns are connected to each other, and the pattern of connectivity is extremely important. So can you see my cursor? Yeah, that's good. So these cortical columns are connected to each other, and this allows them, as I said, to actually communicate with each other. So, for example, you can see this one here that I'm pointing to is connected to a large number of other columns and can actually influence them. So it might activate them, it might even deactivate them. And basically, by sculpting this connectivity, the brain is able to learn to construct a model of the environment, basically, and we'll look a little more detail on that in a second. So the brain.
Dr. Gallimore?
Yes.
Someone asked a question. Do cortical columns have all or nothing behavior similar to a neuron?
Not quite as all or nothing as a neuron, no. So they have different, clear, distinct kind of up states and down states I mean, with a neuron, you've got an action potential, so it fires or it doesn't fire. That is a truly all or nothing system. With cortical columns, which are built from many of these neurons, it's not quite as simple as that. But it's a reasonable approximation or reasonable simplification to suggest that they're either active or inactive, if that makes sense. Great. So yes, so basically you have this pattern of cortical column activation, which is sculpted, if you like, by the connectivity. So the connectivity will determine the sort of which patterns are more likely than others. And that's really what your brain is doing. You know, it has learned to do through evolutionary epochs. Much of the connectivity was established there, but also from the moment you're born, basically, your brain is absorbing information and it's sculpting its connectivity and trying to refine its model of the environment. Okay, so this is kind of where we are so far. So each pattern of columns is a single state of the cortex, which encodes a vast amount of information. And the reason it really encodes a vast amount of information, because when the cortex selects from one state, it's effectively ruling out an almost infinite number of other states. And so that state, each moment of your life, each moment in which you're existing in a world is literally a unique state. You know, you will never step into the same moment again. You will never step into the same river twice, they say. You will never exist in exactly the same moment again, because your brain can create a practically infinite number of moments. In fact, one could say quite reasonably that the moment that you're experiencing now, nobody has ever experienced before, nobody will ever experience again. And that's purely done with activation patterns of cortical columns, which I think is quite remarkable. So your world model in its entirety is encoded by the pattern of activation of these cortical columns. Or we can say that your world model is a highly complex pattern of information. Okay, so let's try and make this a little bit more concrete. So let's imagine we've got this very simple world that we want to encode using cortical columns. So this world is very simple. It's simply the blue circle in this image here. So let's imagine that there are only 4 possible colors. Let's imagine there are only 4 possible shapes. To keep things simple, how would we encode a blue circle? Well, we've got 2 features that we need to encode. We need to encode the shape. So again, let's say there's only 4 possible shapes. So we need to encode the fact we've got a circle here. We need to encode the color. So basically we need, in this case, let's say 8 columns. We will functionally segregate the columns. So 4 of the columns we will say are responsible for representing color information. So we've got a column for the yellow, column for blue, column for red, column for green. Same with shapes. Again, there are only 4 possible shapes in this very simple reality: a square, a triangle, circle, and a pentagon. Now, of course, in reality, in truth, there are many more colors, many more shapes, and there's no way that the brain is able to represent the color yellow using a single cortical column. But this idea of functional segregation, the idea that different columns are responsible for representing different types of information, is the key point here. So how do we represent the blue circle? Well, it's pretty straightforward, pretty obvious. We activate the blue column, we activate the circle column, and then we have a pattern of activation of these 8 columns, which represents this blue circle. So if you were existing in this brain that contained these columns, you would experience this pattern of activation as this blue circle, which is quite difficult to envisage, perhaps. A red square, another possible colored shape. Again, we've got a simple, a different pattern of cortical column activation. We activate the red column, or the column that represents the color red, and we activate the column that represents the square shape. And we can represent all possible combinations of colors and shapes by using different patterns of cortical column activation. Okay, so let's complicate it a little bit. So let's imagine we've got something a little bit more complex than simple shapes. We've got something we might see in the real world, perhaps, or in a cartoon, some kind of little alien face modeled after myself. And this is the perceived object. This is what you experience in the world. And the question is, how does the brain represent the features, the informational structure of this perceived object? What is the cortical representation of this object? Well, we need to think about what are the features that we need to encode. Well, we've got shapes. We've got a number of different shapes. The shapes formed from lines. We've got circles. We've got the mouth contains a number of shapes. We've got the different colors. We've got the texture. Also, this is supposed to be moving, so we've got the movement as well we need to encode. So what the brain does, again, is it functionally segregates different cortical columns to represent different types of information. So there are cortical columns in the cortex responsible for representing movement, for motion. We've got columns for representing color, columns for form, a bit of an oversimplification there, and the same for kind of texture-ish. But basically, the different features of the world are represented by different types of cortical columns. But overall, you will see that this rather complex visual object, this little face, is actually fundamentally represented as a pattern of cortical column activation. So it's a particular pattern of cortical column activation. And so overall, your entire world at any point in time, believe it or not, is represented as this pattern of cortical column activation. Now, in reality, your world has this kind of, if you look around now, you will notice that your world has this kind of hierarchical construction. You look around, you see the scene overall, but then you notice that your scene is filled with objects, and you can name these objects. And then each of these objects you can kind of unpack and take apart and say that, oh, this is I'm looking at, let's say, a tree in the garden, and you can see that the tree is composed of a number of different shapes, large numbers of these kind of ovoid shapes which have a green color. You're looking at the trunk, which is kind of a more linear kind of tubular kind of structure, which has a number of kind of shades of brown or whatever. And so you can take apart and notice that the world, there seems to be this hierarchy from simple features that are kind of put together to form these recognizable objects. Now, there are really no objects in the world as such, but your brain kind of parcelates the world into these recognizable objects, which allows you to make sense of the world. And so we can see that with this little alien face. I haven't completely kind of unpacked this, but we've got the kind of what we would call the higher order structure, the object itself, which we recognize as this face. But then we can break that down and say, well, the face is composed, perhaps we can look, the eyes, the nose, the mouth, these are basic features of all faces. And in this case, the eyes, we can unpack and say, well, the eye is composed from a number of circles, nested circles. There's the color blue, there's the color yellow. So we can break these objects down. And this is really how your brain constructs your reality as this hierarchy of features, from simple features, what we call lower order structure, all the way through objects and these higher order structures. And you can see this in the topography of the visual cortex. So the visual cortex at the back of the brain, of course, responsible for representing visual information, and we'll stick with visual information. Everything I'm saying also applies to the other senses, but we are visual creatures. The psychedelic experience is very much a visual experience, so we'll stick with the visual. So if we look at the visual cortex, we see the first area of the visual cortex called V1. This is really has the closest access to the external world. It's receiving information from the environment via the thalamus, and is really responsible for picking out and representing the basic features of the world, such as lines moving in a particular direction, particular spatial frequencies, basic movement and color information. And then as you move up the cortical hierarchy, V1 sends its information to V2, which sends it to V3, to V4, to V5, etc. And then at these higher levels of the visual cortical hierarchy, we could call it, you start to, all of these lines and then shapes and then colors, and then they're put together, and then they form these higher order structures that we recognize as a face, for example, or a tree. So this is kind of the visual pathway as it's understood. The information comes from the retina, goes to this is a part of the thalamus that's responsible for transmitting visual information, goes to the back of the cortex, the V1, and then passes up this hierarchy. And there are two pathways here. Don't worry too much about the importance of these, but the idea is that you're going from a lower order to a higher order. And all of these parts of the hierarchy are equally important in constructing your world model. Okay, so let's kind of update our picture of the cortex. So we will kind of include now, so we'll imagine that this is, again, it's obviously massively oversimplified, but this is what the cortical hierarchy might look like. You've got the lower order, let's say here on the left-hand side, kind of medium order, and then higher order. So the lower order, this is representing, lines and spatial frequencies, basic information, and then right at the other end, this higher order is where we get things like objects are picked at. So this is our representation of the visual cortical hierarchy. So the usual way that this is thought about, the classical way, if you like, is that sensory information is kind of driving, your brain is kind of absorbing all this sensory information and using it to construct your world model at each moment. So your sensory information comes from the, let's say, the retina via the thalamus. It stimulates certain cortical columns based upon what you're looking at, based upon the information it's receiving, picks up certain lines in certain orientations, moving in certain directions, puts that together, passes that up to the next level in the cortical hierarchy, which puts together some of that information, and then finally passes that information up to the highest levels of the cortical hierarchy, where you get the appearance of you're looking at a face, you're looking at a cat or a dog or whatever. So this is kind of what's often referred to kind of as a bottom-up sensory information driven way of looking at perception. Your brain is constantly having to absorb this information. However, this is kind of a little bit outdated now, and we think about perception kind of differently. We think about the way that your brain constructs your world model in a different manner. So this is the old school bottom-up information flow approach. Now, in truth, what we think is happening is that rather than kind of relying on sensory information, basically at any point in time, your brain has a model of the world. Your brain has a set of hypotheses, best guesses about what it thinks is going on in the outside world. What's the content of the outside world? Who is out there? What is out there? What are they doing? What is happening? Everything that's going on in the world, your brain kind of has this picture, this model, and that's what you're experiencing. You're experiencing this model, your brain's best guess about what's going on in the world. And based upon that best guess, that model, your brain then makes predictions. Your brain says, okay, this is what I think is going on in the world. If that's the case, what kind of sensory information should I be receiving next? And so your brain is a prediction machine. It doesn't rely on sensory information as such in order to construct the model. It already has a model. What it does is it tests the model using sensory information. It says, okay, if this is true, if my model is good, I should be able to predict the next pattern of sensory information that my eyes are going to receive, for example. We'll look at examples of this to make this clear, but basically this is the idea. So to illustrate this, this is a kind of a famous illusion, which you may have seen before, but if you have seen it before, that's fine, but we're going to think about this in a slightly different way. So at the moment, you're looking at a face. Now, your brain has instantly, your brain is very good at, knows a lot about faces. It has very good models of faces encoded in its architecture, in its cortical architecture. So when your brain is looking at this face, it sees a normal face. Now, that is a regular kind of convex face. And, this is your model. Your brain is settled on this particular model of the face. Now, based upon that model, as the face starts to rotate, which it will in a second, your brain is basically starting to make predictions. It's saying, okay, if this is just a regular face, then it should very, very easily be able to predict the patterns of information that it's going to be receiving as the face rotates. So let's, for example, it should start to see the nose coming to profile, the whole kind of rounded structure of the face. All of this should happen. And so the brain is expecting this and predicting this. However, when you actually start to see the rotation, you can start to see that, in fact, this is not a regular convex face. It is the other side of a mask. However, your brain is so used to seeing regular convex faces that even now, even though you know that this is the other side of a mask, your brain still has this model. And what's the important point here, which I think people often miss, is that when you're looking at this face, even though we know it's a concave face, your brain is still constructing a convex model. So in a sense, the model that your brain is using is exactly the same as the model it would use if it was looking at a convex face. It's using the same model. It's decided this is a convex face, and so it's actually constructing the convex face model. However, as soon as it realizes that, basically its predictions start to fail as the face starts to rotate, the brain realizes, oh no, I'm not seeing the right pattern of information. I'm not seeing, as we're seeing now, this nose coming to profile. So my model must be wrong. My predictions are not being fulfilled. So your brain is really doing that all the time. It has a model, it makes predictions based upon that model, and then it's testing its model against sensory information. So your model is really constructed at a very high level. Your brain has decided there's a face in the world. It's decided there's, some other object, some ball rolling down a hill or whatever. It's decided this is what's going on. And based upon that, it says, okay, now I'm going to try and predict the sensory information. And it does it in a stepwise level, stepwise manner, working down the cortical hierarchy, so that the higher level model of what's going on in the world leads to predictions about the activity in the lower order. So let's, in this case, this kind of middle order, medium order area of the visual hierarchy. So the top level is essentially predicting the pattern of column activation in the medium order. And then the medium order in turn is attempting to predict the column activation in the lower order, and the lower order is itself trying to predict the patterns of sensory information that it's going to receive in the environment, from the environment. And sometimes it gets that right. If it gets it right, and this is really the beauty of this way of working, if it gets it right, basically the brain knows that its model is working. It knows its model is good. So it doesn't need to know about this sensory information. And so it will basically just quash, it will inhibit it. It will extinguish that sensory information. It will filter it out. It will say, I don't need to know this. Because processing sensory information is expensive. You have to generate all these action potentials. You've got all these synaptic connections. This is expensive stuff. The brain uses a lot of energy. So if the brain was just to absorb all this sensory information from the environment, it's a very expensive process. And all of this sensory information also has the potential for kind of perturbing the brain and causing it to become kind of disordered, disorderly activity. The brain is always poised between order and disorder, and it's a careful balance. So the brain doesn't want to be absorbing sensory information that it knows about. So if the brain successfully predicts this pattern of sensory information from the environment, it basically extinguishes it. It doesn't get passed up the cortical hierarchy. So the brain is basically always trying to do this. Every moment the brain is trying to predict the next pattern of sensory information and extinguish it. Imagine there's a wall, a dam, and there are holes in the dam, and your job is to try and stop the water from flowing out, lest the whole village should be flooded, right? And you have to learn about, what are the patterns of water coming out of these holes in the dam? Can I predict them? Can I stop them? And that's your brain. Your brain is kind of fighting all the time to kind of stop this sensory information from overwhelming it. And it does it by learning to predict the sensory information. And it predicts the sensory information by having a good model of what's going on out in the external world. Is that clear? Good. Okay, so what happens when it gets it wrong? Now, sometimes the model isn't going to be perfect at all. This is a rolling model. And so sometimes the sensory information will be wrong, such as when that hollow face rotated, the predictions start to fail, right? So here we got a good match between the prediction and the actual sensory information. We extinguish that sensory information, great. Sometimes though, the model fails to predict the sensory information. Then you get a mismatch, a discrepancy. And this generates what are called, suitably enough, reasonably enough, prediction errors. And this is the information the brain needs to know about. If its model is working, it doesn't need to know about the sensory information. It doesn't need to know about predictable sensory information that it can successfully predict. But surprising information that it didn't predict, that's what it needs to know. In other words, it needs to know about these prediction errors, the mistakes it made. And so it's these prediction errors that get passed up the cortical hierarchy. We get a mismatch between the sensory information and the prediction. This generates the prediction error. The prediction error flows up the cortical hierarchy and is then used to refine the model, to update the model. So we had this original model. Again, it failed to predict the sensory information. So then we update the model, from the level of the highest level of the cortical hierarchy down, and then hopefully this updated model here, brilliant. We successfully predicted this sensory information now. So the model has been refined, and this is happening all the time. This is a rolling process. This doesn't kind of happen every 10 seconds. Your brain kind of takes some information and updates. It's happening all the time from every moment of your life. So let's kind of take stock a little bit. So your this high-level model generates predictions about sensory information, and these predictions are passed downwards down the cortical hierarchy with the aim of predicting the sensory information. If we get a good match, that sensory information is extinguished. The brain doesn't need to know about it. But any errors are then passed upwards through the cortical hierarchy. So most of the information, in contrast to the kind of classical view, most of the information is actually traveling down the cortical hierarchy in the form of these predictions. We'll skip that for the time being. Okay, so this is basically what your brain is this is basically how your brain is constructing your world. So your brain generates a model of the world using information. Now you understand exactly what I mean by that and how that happens. The model is used then to make predictions about sensory information. The predictions are then compared with the actual sense information, and the discrepancies generate prediction errors. The prediction errors then are passed up the cortical hierarchy and used to actually make small refinements, updates to the model, and then restart that cycle again. And this is happening all the time. Okay, now, so let's think about dreaming. So when you are dreaming, your brain is actually constructing the world in pretty much the same way as it does when you are awake. The key difference is not in the way the world is constructed. It's still constructed using these pattern of cortical column activation. But with the difference is that these primary sensory areas at the back of the brain are, well, not so much shut down, but they're disconnected from the environment. So the relays from the thalamus that normally connect the outside world to the primary sensory areas, the primary visual cortex particularly, are kind of shut down. And so the brain loses its connection to the environment, but it's still perfectly capable of building a model. So when you're awake normally, this model, as we've seen, is being tested all the time against sensory information, and that keeps it stable. That keeps it a coherent, stable model. When you're dreaming, your brain is still perfectly capable of constructing the model. You know, your dreams are not kind of hazy suggestions of a reality. They are practically indistinguishable from the normal waking world in many aspects, and studies have shown this. All of your senses are intact, despite what you might believe. And you know, the amount of time you spend talking on the telephone or watching TV or whatever is normally pretty similar in the dream world as it is in your waking world. But the one thing about dreaming is that it often can become strange. The scenes can shift. The identity of people you're talking with can often change. You know, one second you're talking to your mother, and then you're talking to your girlfriend, and you know, this kind of horrible juxtaposition of people. And it's but you don't recognize it for what it is, because there are also these frontal areas of your brain that normally would critique that situation and say this is impossible. They're also kind of inhibited as well, unless you can learn to lucid dream, which you kind of able to kind of reactivate those frontal areas of the brain. Then you can recognize that you're in a bizarre dream and control it. Anyway, the key point here is that during dreaming, this model testing isn't possible. So sensory information is a training signal. It's designed to allow this model to be tested, which keeps it stable and predictable and coherent. Okay, so now, any questions so far, by the way, before we actually talk about the effect of psychedelics on this model?
We're having a whole debate as to whether or not something like motion sickness would constitute a prediction error, wherein you're perceiving external stimuli that is relatively static, but you're feeling motion. Is that something that your brain would say, you know, based off of my past experiences, I shouldn't be experiencing this?
Or is it like when you miss a step going down the stairs because you perceive it to be closer or farther away than it really is?
That's a very good one. So with the motion sickness one, I think that's a little bit complicated, but I think yes. Or when you get off a boat and the world still keep moving, you know, because whilst you're on that boat, your brain is adjusted. Your brain knows now. It gets used to the movement of the boat. And so so that's why you stop feeling it. You stop feeling the movement. Your brain is predicting that now. And so it's like you're standing on just terra firma, right? As soon as you get off the boat, your brain is still predicting and you're still experiencing then you get this jarring effect where you get this discrepancy between the predictions, and so for a while it's like you're still on the boat, and you're actually feeling the movement now more than you actually did on the boat itself because of that discrepancy. And it's those prediction errors that you start to experience. And with motion sickness, similar idea, I think, yeah. But certainly, yeah, with a step, that's when you really notice it, right? Because you've got, and it's not just the visual here. You've also got your proprioception. So your brain knows where the position of your limbs. It knows expecting that firm touch on the base of your foot. When that doesn't happen, it's jarring. or picking up an empty suitcase is the one that comedians in the 80s used to get a good laugh out of. Have you ever picked up a suitcase and thought it was full? That thing, right? That is the same idea, because your brain, it sets up the musculature to pick up the suitcase, and then it's empty, right? So yeah, so you can see it. You can experience the effects of this, the brain's predicting, and that's absolutely vital. You know, your brain couldn't function. You certainly couldn't construct a rich and complete world if your brain had to constantly had to kind of construct it from scratch, so to speak, in this bottom up fashion using sensory information. So the brain uses as little sensory information as it needs, and it's constantly making these predictions.
Thank you.
Thank you.
Okay, so now we get to psychedelics and the effect they have on the world model. So using this kind of updated way of thinking about the way the world model is constructed, we can really get some really nice insights into how psychedelics work. So we're going to focus on the so-called classic psychedelics. So those are the ones that are loosely based on serotonin. So serotonin in the center here, you can see. And then on the left, we've got psilocybin from magic mushrooms. Psilocybin, of course, not quite the active component. Psilocin is when you remove this phosphate. Basically, that's the compound you find in the magic mushroom. And you've got LSD, of course, perhaps the most famous of all the psychedelics. So first of all, the problem. The problem that we have in trying to understand psychedelics. So far, when we're talking about the world model, we're speaking at quite a high level. We're talking about the activity of cortical columns and kind of this emergent patterns of complex activity that represents your world model. When we're talking about psychedelics, we have to start at a much lower level. We have to start thinking at the level of molecules and receptors. Right, and this is why understanding the effects of psychedelics is really quite difficult, because we have to work at different levels of organizational complexity. So, as you may have studied or thought about, the world, just like your world model is hierarchically constructed, so is kind of so is really reality, in a sense, in that you have fundamental particles form atoms, and atoms form molecules. And molecules, large numbers of molecules, then interact with each other and form these living structures that we call cells. And so this line between the gray and the blue here represents the kind of moving from the inanimate or the non-living to what we would truly call a living system, cells, basically. And then cells form networks. They connect and interact with each other to form tissues, then form organs, and then form the organism. And so a cell itself, just a single cell, such as a neuron, for example, of course, the brain is built from large numbers of neurons. So the brain is a complex, highly complex system that functions, has emergent functions that emerge from the multitudinous interactions of billions of neurons. That's in itself is complicated or complex in the truly complex scientific sense. It's complex, but each neuron itself is also a complex system. Each neuron is, if we look at the internal milieu of a cell, you can see that don't try and understand this. You have a myriad of interactions between molecules, and it is from the interactions of these molecules that the emergent function and behavior of the neuron emerges. So, generation of the action potential, maintenance of the membrane potential, the production and repair, replacement of proteins, copying DNA, dividing in some cases. These are all emergent properties. And so what psychedelics do, which makes them really difficult to understand, is that they perturb this highly complex system in a very specific way, the neuron, which causes a subtle change in the emergent behavior of the neuron, which then affects which then perturbs this highly complex system called the brain, which is generating your world model. So you've got complexity at a number of different levels that you need to reckon with. And complex systems are, part of what defines complex systems is that often very small changes can have quite dramatic effects on behavior. And so very, very small changes at the receptor level, which we're going to look at, can have very dramatic changes on the structure of your world model, of neural activity at the cortical column, networks of cortical column level. Yeah. So anyway, so there's the problem, and we're far from kind of unraveling this system, and this is where modern systems biology comes in. This is basically what I do for part of my my work. I'm a computationalist. I'm a theoretician, so I try and put these systems of complex interacting systems of molecules into a computer, try and model their interactions, and then look at the emergent behavior. And that's really what you have to do to try and understand these complex systems. You can't simply look at a piece and say, oh, this does this, this does this, this does. It's not a car. This is not just a machine. It is a emergent complex system. Okay. Anyway, so there's the kind of the problem that we have to deal with. But we have made a lot of headway in the last few decades and how this all works. So this is kind of a classic picture of the way receptors work. So psychedelics, they bind to receptors, and we'll look at specific examples of the receptor that psychedelics bind to and how that works. But the basic picture is that a molecule binds to a receptor, and two things can happen. Either nothing happens, it doesn't activate the receptor. We call this an antagonist. So this red molecule is binding into the receptor, nothing's happening. All it's doing is blocking other molecules, perhaps potentially from binding. On the other hand, this blue species here, this blue molecule is binding to the receptor. It's binding in the receptor, the ligand binding site on the outside of the receptor. So this is kind of, yeah, this is, I should draw on this in, but anyway, you've got the membrane here. So this is the outside of the cell, this is the inside of the cell. So this blue molecule, which we call an agonist, is binding to the receptor. It distorts the receptor in some way as it binds and interacts, and causes a conformational change in the protein, which is transmitted in a sense through to the intracellular domain here, which kind of changes its shape and allows it then to interact with signaling molecules on the inside of the cell. And if you read that chapter that I sent, that PDF, you will have seen examples of this kind of signaling. And that's the basic principle, the idea that receptors really, they are transmitting information from the outside to the inside of the cell using distortions of the receptor shape, the receptor conformation. And that's basically what psychedelics are doing. They're shifting the receptor from the inactive to the, this is kind of the inactive state to the active state, which causes interaction with signaling molecules on the inside, which then causes the specific effect on the neuron's function and behavior. So we can see this particular example. So this is actually a dopamine receptor. Now, this is from a modeling study, a fairly recent modeling study. And you can see here, this is what's called the apo state. So this is an unbound state, nothing's bound. And then as we move to the left, the agonist binds to the receptor, and you can actually see, if you look at the receptor, you can see that it's distorted its structure. And in fact, it's distorted its structure such that it can then bind to this large signaling protein here called a G protein. And the G protein then can interact with one or more signaling proteins and generate the effect in the particular neuron. And so part of what we have to do when we're thinking about psychedelics is learn to try to understand what is that effect, what's actually happening when we activate the particular receptor. We need to know what receptors these molecules bind to, psychedelic molecules bind to, and then we need to know about what is their effect. So some of you will know that the psychedelics, they bind to a particular type of serotonin receptor. receptor. So serotonin is 5-hydroxytryptamine. And in particular, we have known now for several decades that the major locus for the psychedelic effect, the absolutely critical receptor, is this 5-HT2A receptor. And we know a lot about what the 5-HT2A receptor does. So we'll just go through this signaling pathway and try and explain what's actually going on here. So the agonist binds, so this red circle is the agonist, so it might be serotonin, it might be LSD, it might be psilocin or DMT. It binds to the receptor. It causes a conformational change, which allows the receptor to bind to this large signaling protein called GQ. It's a G protein. And the G protein then interacts with and activates an enzyme called phospholipase C, PLC. It's a lipase. It breaks down this fatty molecule called PIP2 into two molecules, two very, very important signaling molecules that you will see all over the place if you study any biochemistry for any length of time. One is called IP3, and IP3 binds to receptors on the endoplasmic reticulum and causes a flood of calcium into the cell. So the concentration of calcium increases dramatically, and the calcium then activates or partially activates a kinase called, very important kinase called protein kinase C. DAG, which is also produced, also activates protein kinase C, and they have this kind of synergistic effect. Protein kinase C is a very complex, complicated kinase, and its activation is quite complex. But DAG and calcium act synergistically to activate protein kinase C. So it's a kinase, so it's a phosphorylating protein. And then through a series of steps, which I've kind of missed out here for simplicity, ultimately this other kinase is phosphorylated. This is called ERK, ERK, sometimes called MAP kinase, another very important signaling protein. And what ERK does when it's activated indirectly by PKC is it phosphorylates potassium channels. And when these potassium channels are phosphorylated by ERK in response, so effectively what's happening is by activating the 5-HT2A receptor through this complex signaling pathway, ultimately it leads to the closure of these potassium channels. When the potassium channels are phosphorylated, it distorts their shape and causes them to close. Now, this is a bit of a test. Can anyone tell me what's going to happen to the membrane potential? Is that too much of a test for you? What's going to happen to the membrane potential if you close large numbers of potassium channels in the neuron? Okay, so what is the, ignore that. What is the distribution of potassium ions across the neuronal membrane? What's the concentration gradient? Are we higher outside or inside the neuron?
Outside the neuron.
Other way around. So sodium is a higher concentration outside. Potassium has the opposite. So potassium has a higher concentration inside. So potassium is constantly trying to flow out of the cell. And there are channels that are variously open and closed that basically allow potassium to flow out. And this is partially balanced by sodium that flows in. You've also got chloride as well, et cetera, et cetera. So it's this flow of channels, this flow of ions that's basically maintaining the membrane potential at all times, the so-called resting potential. So when you close the potassium channels, so potassium normally it flows out, it takes its positive charge with it. So potassium channels, when potassium flows out of the cell, it's essentially it's tending to push the membrane potential in a negative direction, because potassium is basically taking positive charge with it. So when you block the potassium channels, when the potassium channels are inactivated by ERK, you actually cause the membrane potential to rise. It becomes a little bit more positive because you're trapping more of this positively charged potassium inside the cell. Does that make sense? It's a little bit, this is something you need to work through. But normally the membrane potential sits at this resting potential. When you block the potassium channels, you're keeping more positive charge basically inside the cell, which pushes the membrane potential slightly in the more positive direction. So that's really the effect of these psychedelics overall, is that they're pushing the membrane potential a little bit higher towards the threshold. And we all know, of course, that the threshold potential is the potential at which a neuron fires. So this is called making the neuron more excitable. You're exciting the neuron. You're making it more likely when the neuron receives these excitatory inputs that are kind of pushing its membrane potential upwards. You're making it more likely that it will reach the threshold potential and fire an action potential. So when you raise the membrane potential of neurons like this, you tend to make them more likely to fire and to keep firing for longer. And this happens with over large areas of the cortex, and we're going to look at the effect of that in a second. Okay, so I'm going to skip this actually. Okay, so let's think about the actual interaction before we kind of look at the effect of this, the closing of these potassium channels. We need to look really at how these molecules bind to receptors, because you're told, or you would have been told if you've been studying pharmacology 20 years ago or 30 years ago, you've been told that an agonist activates a receptor, an antagonist doesn't activate the receptor. But what we know now actually is that the way that a receptor is activated, the way that a molecule will distort the receptor and change its conformation will vary depending upon the molecule, depending on the structure of the molecule. And this will affect how that receptor then interacts with signaling pathways on the inside of the cell. So to take an example, we've got serotonin on the left-hand side here. Serotonin has a certain shape. It has certain functional groups, as they're called in chemistry. So it has this amine group, this nitrogen group here. It has this hydroxyl, this oxygen hydrogen group, this OH group, and it has this overall shape. DMT has a similar shape, but it's not quite the same. It doesn't have this hydroxyl group here that the serotonin has. It has these two additional groups on the nitrogen, these dimethyl groups, right, which gives it its name, dimethyltryptamine. So serotonin and DMT, they're both going to bind to the 5-HT2A receptor, but they're going to bind in a slightly different way. They're going to sit in the receptor slightly differently. They're going to interact with different parts of the receptor, and they're going to distort the receptor in a slightly different manner. And we now understand that to be really important for the way that psychedelic drugs work. So, for example, on the left-hand side here, we've got a drug that looks nothing like either DMT or serotonin. It's actually more closely related to mescaline. Anyone actually know what this is? It's a very famous psychedelic. It was invented by Alexander Shulgin. This is 2CB. The B stands for the bromine here. So this is a family of psychedelics. He also created 2CI and 2CC, the iodine and the chloro equivalents. But this is a very popular and very potent psychedelic. It also, it works at the 5-HT2A receptor, but it has different effects. It's not the same as taking psilocybin or say taking DMT. And this is kind of an interesting question. Why is that? And part of it can be put down to the fact that this, whilst binding to the same receptor, it's actually binding to the receptor in a slightly different way. So this 2CB, it lasts, normally for 4 to 8 hours, sometimes longer. Normally you take about 1 milligram, 1 to 10 milligrams. and it has very distinctive effects. This on the right here, this is called bromo dragonfly. This was invented by Dave Nichols. I think he's now retired. He was at Purdue, one of the most important medicinal chemists, psychedelic medicinal chemists in history. And this, again, very, very different kind of shape. You can actually start to see that what they've done here is kind of folded this this group round to form this kind of winged structure and given it a slightly different shape using what they learned about the receptor structure, and you get a very different drug, a very potent drug. You only need about 0.2 milligrams, and it hangs around in the receptor for a very, very long time and has quite potent effects for, often for more than a day. So this is not a drug to be messed with, so to speak. LSD. So this was from a paper that came out a couple of years ago. LSD is kind of, everybody knows that LSD is extremely potent in that you need very, very small amounts. It binds the 5-HT2A receptor. You don't need much, 50 millionths of a gram is kind of an active dose, 50 micrograms. We now know, and the paper was actually just published a few weeks ago, that kind of verified this, that when LSD binds to the 5-HT2A receptor, it kind of sits in the receptor in such a unique configuration that it actually causes the receptor to kind of fold over the binding site and form what they refer to as a lid structure. So it sits in the receptor, and then a lid folds over, and that really keeps the LSD in the 5-HT2A receptor for extremely long periods of time. So that's why it doesn't kind of wash out. It isn't removed and available for degradation and metabolism by enzymes, basically. So that's why we can understand now why LSD lasts for such a long time. It's because it sits in the receptor for such a long time. So you don't need to give a large dose of LSD. So this old picture of agonism versus antagonism, either activates the receptor or doesn't, is now outdated, and we now have to think about how the molecule sits in the receptor to really understand the effects of the different effects of drugs. So all of these classic psychedelics, they all bind to the 5-HT2A receptor, and yet they all have quite subtle effects, and that's partially to do with how they bind to the receptor. So what we know is that something called functional selectivity. So these are both agonists, but they both bind to the receptor in a slightly different way. They cause a different conformational change in the receptor, which means that the intracellular domain develops a slightly different conformation in response to the agonist binding, and that allows it to interact with different signaling pathways. So this really complicates things even more. There's also the issue that drugs sometimes, drugs rarely bind to just one receptor. They will also often bind to other receptors, other serotonin receptors. And so you've got each receptor is kind of perturbing this complex network of molecules inside the cell to generate this particular effect. We saw one effect, the closing of the potassium channels, but then you have the molecule, the drug can also bind to other serotonin receptors, which have other effects. And so you have interactions between these signaling networks, and then it gets very, very complicated. It's often very, very difficult to predict what is the effect of a particular psychedelic molecule going to be on a single neuron. That's difficult to predict. And then you have to predict what's the effect going to be on the structure of your world model. What kind of psychedelic effect is it going to have? Very difficult stuff, I'm afraid. Okay, so let's look at a particular example that kind of illustrates this. So we've got two molecules, LSD and glyceride. They look very, very similar. In fact, they are very, very similar. There's very little difference between them. The only difference really is that with glyceride, you've got this nitrogen added kind of in between these two carbons. Now, LSD, as we know, is highly psychedelic, highly potent. Glyceride is not psychedelic. It is, however, an agonist at the 5-HT2A receptor. So what's going on? We thought that agonists at 5-HT2A receptor should be psychedelic, right? Well, no, of course not. Serotonin is not psychedelic. And yet it, of course, is the natural agonist, the natural activator of the 5-HT2A receptor. So there's clearly something going on here. It's not just about activating the 5-HT2A receptor, but about activating it in a certain way. Now, this study came out a few years ago. This is a modeling study which computationally docked different molecules into the 5-HT2A receptor and noticed that there was a distinct difference. So when and they were looking at a particular part of, this is the intracellular part of the domain. You can see the membrane here. And so when the non-psychedelic agonist binds to the receptor, it activates the receptor, but what it doesn't do, it doesn't flip this little loop. They noticed this little loop in the protein structure kind of remained still when something like glyceride or serotonin was docked into the receptor. However, when a psychedelic molecule, they use DOI, which is another one of Shulgin's creations, but when a psychedelic molecule binds to the receptor, it causes this loop here shown in blue to actually flip outwards. So we can clearly see here that there's a difference in the way that psychedelic molecules are distorting the receptor, changing the conformation of the receptor versus non-psychedelic molecules. And this is the first clue as to what's going on. So what's actually happening then? Well, it turns out that the 5-HT2A receptor is not the only receptor that's important in psychedelic drug action. And there's another critical type of receptor called a glutamate receptor. A glutamate is one of the most important neurotransmitters, if not the most fundamental and important neurotransmitter in the brain. But there are many types of glutamate receptors, and one of them is called mGluR2. Now, what both of these, mGluR2 kind of works in the opposite way. It does the opposite to what the 5-HT2A receptor. So we looked at what happens when you activate the 5-HT2A receptor. Via this pathway, which I've missed out here, it inhibits the potassium channel. It phosphorylates it and it causes it to close. It deactivates these potassium channels. The glutamate receptor, on the other hand, via a different signaling pathway, which again I haven't shown here, actually does the opposite. It activates. So the 5-HT2A receptor is kind of fighting against the glutamate receptor. The glutamate receptor is trying to open these channels. The 5-HT2A receptor is trying to close them. And it turns out that when you add something like glyceride, it's not psychedelic, not because it's not activating the 5-HT2A receptor, but simply because you get this kind of counterbalancing effect of the glutamate receptor. So any effect of the 5-HT2A receptor is kind of cancelled out. It's not strong enough to actually cause a psychedelic effect. However, when you use LSD or any other psychedelic drug, as we saw, it induces a slightly different conformation in the receptor, which actually allows that 5-HT2A receptor to stick to these glutamate receptors and actually inactivate them. When that happens, basically, it means that this glutamate receptor stops working. So it stops being able to kind of oppose the 5-HT2A receptor. So these psychedelic molecules, they're doing two things at the same time. They're activating the 5-HT2A receptor pathway, but they're also inactivating another pathway in an entirely different receptor. And both of these are necessary for the psychedelic effect to occur. Okay, so now, final bit. Let's kind of put this together now. So we understood, so at the start, we were talking about how the world model is constructed. Then in the second part, we looked at what the psychedelic molecules are doing. They are depolarizing, they're exciting the neuron, they're raising the membrane potential of neurons, making them more likely to fire. Now, which neurons are we actually talking about here? Well, we're talking about a specific type of neuron in the deep layers of these cortical columns. Now, remember, these cortical columns, they have a number of layers, 1 to 6. So these, as I've shown here, that I'm just circling around, these are called pyramidal cells. They're called pyramidal cells because they have this pyramidal structure. You can see the cell body kind of sitting here that I'm pointing to now. You have this long dendritic tree. This looks like an axon. It's not. These are the dendrites. These dendrites reach up to the highest levels of the cortex, the highest layers 1 and 2, etc. And then you have an axon you can't really see here, you can't pick out, which then allows this pyramidal cell to communicate with other columns. So each column contains a large number of these pyramidal cells. And these pyramidal cells are absolutely critical in constructing your world model. And they're also important in allowing columns to communicate with each other. So let's look at this a little bit more schematically. So we've got 3 columns here. I've separated the layers. So we've got these kind of the layers 1 to 3, kind of, we often call these the superficial layers or the shallow layers. And then you've got the yellow part here. These are the deep layers, so kind of 5 and 6. And this is where we find our pyramidal cells. They're called pyramidal cells because they're shaped kind of, the cell body is kind of this pyramidal structure. And so we can see the cell body of the pyramidal cell. And the pyramidal cell is important, as I say, in activating a particular cortical column. And of course, that's important in constructing your world model, because your world model is a pattern of activation of these cortical columns. So these pyramidal cells communicate with each other. So you have the axon here. So this pyramidal cell is, again, in a highly simplified fashion, is communicating with part of the dendrites of another pyramidal cell. And this is how, basically, in an extremely oversimplified sense, columns can actually speak to each other. So columns speak to each other. Your brain, your cortex, sculpts its world model by controlling the connections between the cortical columns. So, for example, so let's say we will take, I've got a few columns here, 12 columns, and we can see with the columns are connected to each other. We've got 3 of them are active, and we've got some strong connections, which I've illustrated with solid lines, and then we've got weaker connections. So as these columns are activated, they're more likely to communicate, to send information to columns to which they are more strongly activated. And so you get kind of completion, if you like, of this particular pattern. And this is really how the brain works. It controls the connectivity, the strength of the connectivity, and that allows it to control the flow of information between columns. So how columns speak to each other and communicate with each other. And that's critical for your brain to construct a coherent, stable model of the world. So now then, let's add LSD. So when we add LSD, we now know what's going on. We know that it's stimulating these pyramidal cells by activating the 5-HT2A receptor, by closing these potassium channels. So all of the pyramidal cells are sitting at a slightly higher membrane potential. They're more likely to fire. And what that means is that normally the weakly connected pyramidal cells won't speak to each other. But in the presence of LSD, you start to get communication between columns that wouldn't normally communicate with each other, because they're more likely to fire and they're more likely to become activated when they receive information from another column, because they are kind of in a more excitable state. And so this kind of rigid structure of the world that's normally imposed by the connectivity starts to break down. So before we saw this, this beautiful shape that's constructed as part of the world model, now we start to see that breaking down. So your world model starts to be disrupted. It starts to fall apart. It starts to become more fluid. The identity of objects in the world starts to change. And so this is something that really I predicted, I guess, back in 2013. This is what was going on in the brain, is that the normal patterns of activity that are responsible for your world model start to break down. You start to get these novel patterns of activation occurring in the presence of a psychedelic drug. You get an increase in what's called the repertoire of activation patterns. And so in a way, you get a sort of democratization, I call it, of cortical columns, where this strict architecture of connectivity that normally controls the flow of information starts to break down because of the increased excitability, and you start to get more random activity, information flowing outside of these normal networks. And this was actually demonstrated by Robin Carhart-Harris's group a couple of years after that, and he'll probably talk about it next week, an increase in what he called the repertoire of brain dynamical states during the psychedelic experience. And in particular, they saw that particular patterns of activation of certain brain areas were only found in, this was psilocybin in particular, only found in when someone was given psilocybin. So you're actually starting to see now how you can connect what's going on at the receptor level to actually change the changes that are actually observed in MRI, functional MRI scanners, which I think is pretty cool. Okay, so let's very, very finally, I've gone on for quite a long time, but anyway. So basically this world model starts to break down. So what happens? Well, it means, of course, your brain becomes less capable of making predictions. If it can't construct a stable model of the environment, it loses its ability to make predictions about what's going to happen next. And that means, of course, it loses the ability to filter out this sensory information. Remember, successfully predicted sensory information is extinguished. If your brain can't make good predictions, you get an increase in these prediction errors. And so the amount of information flowing into the brain that successfully flows into the brain and works its way up the cortical hierarchy starts to increase. So the world, not only does the world suddenly start to change in its structure, but it suddenly everything becomes surprising. Your brain feels like it's overwhelmed with information because your brain is literally losing its ability to predict and thus filter sensory information. So you get this barrage of information coming in, of novel, surprising information. So everything becomes fascinating and new and salient. Okay, so that really gives us two of the key properties of the psychedelic state from this. By stimulating the pyramidal cells, your brain loses the ability to construct a stable world model. Information starts flowing out of these normal normal kind of patterns of cortical connectivity, and the world shifts from being this very kind of stable and predictable world to being a very fluid and unpredictable one. This causes an increase in prediction errors. The brain is unable to filter the information because it's unable to predict it, and then information starts flowing up through the cortical hierarchy, experienced as a world that's much richer, full of more information, more interesting and surprising information. Another effect is that we saw before that when the brain fails to make predictions, it attempts to update its model to try and get rid of these prediction errors. The brain does this as well under the influence of psychedelics, but it fails. It constantly fails. And so if you've taken a good, decent dose of a psychedelic molecule, you will find that the world becomes unstable. You will look at an object and it will change its identity. You know, you think you're looking at a wine bottle, and then it suddenly turns into a dog or something like that that's staring at you, and then it changes back again. You know, the brain is kind of shifting from hypothesis, best guess to try and reduce these prediction errors. But it fails, of course, because its model is kind of disrupted. So I think what's neat about this is that we can kind of explain what I think are the most pertinent features of the psychedelic state, certainly in terms of the structure of the world model, using this idea of this what's called predictive processing model of how the brain constructs your reality, and then relate that to what's actually going on at the receptor level. So I think I will take a break there, stop there, and then we can talk about, if you want, we can talk about DMT. I can answer questions. I will leave it up to you.
I mean, that was awesome. Thank you so much. I mean, personally, I would love to talk a little bit about DMT, but I also want to open up the floor to Lexi and Lauren in particular, because they prepared a couple questions in advance of today. Mm-hmm. So if either of you two have any questions that you want to ask, let's do that now, and then see where it goes from there.
Okay.
Hi, we can try and like shorten it slightly so that maybe we can still get to some stuff. on DMT. I can go first maybe, and then Lauren can ask one. So one of my questions was that in the essay you shared with us that you wrote, you talked about how people on DMT were like seeing the same things as each other. So I guess that actually is about DMT anyways.
Yeah.
And I was wondering if you could go more into detail about what they were seeing and like how they were reaching the same place.
Right. Okay. Great. That's a perfect question because it brings me in basically what I was going to talk about. So DMT is special. You know, I call myself a DMT exceptionalist in that DMT is quite remarkable. You know, what I've described so far. Let's see if I've got the right. Yes, this is the slide. So what I've kind of described so far is what regular psychedelics do at regular doses. So this is what happens if you take a couple of dry grams of mushrooms, you take 50 to 100 micrograms of LSD, 300 or 400 milligrams of mescaline, this kind of thing. This is the kind of effect that you're going to achieve. But what and it's this kind of I sometimes in the essay I call it like a nudging the world out of tune, if you like. You're disrupting your brain's ability to tune into the environment. All of the kind of the wonderful effects that brings about. Your brain's ability to see the world anew, in a sense, something you haven't really had the ability to do since you were a young child. So psychedelics are great in that respect, but DMT is different in that it seems to switch the reality channel entirely, rather than generating a more kind of labile and fluid and unpredictable and novel version of this world. You are completely, this world completely disappears. And what that means, and this is the fundamental point, is that your brain is constructing an entirely new model of reality. It's constructing an entirely new world. It's no longer constructing a model of the environment or this environment, the normal consensus world, waking world, normal environment. It's constructing an entirely new model. It has to. That much we know. A lot of people don't appreciate that because it's very common in psychedelic circles. People say, is it real or not? But that's not a very good question. What you really should be saying is whether it's real or not, the DMT world is it's being constructed by your brain. Your normal waking, as we just saw in the lecture, when you're in normal waking life, your world is being constructed by your brain. That doesn't mean that when you go to the park, the trees aren't real. It simply means that your brain is building a model of the environment and is using sensory information from the environment to inform and test that model. So asking whether the DMT world is real or not is not really the question. What we should be asking is it entirely constructed by the brain without access to any sensory information? So there's no model testing. I've often described this as like a dreamt DMT world, a world that the brain is making up of its own accord, or is it a sensed DMT world in which the brain is actually testing this model against sensory information, but it must be sensory information from somewhere else. Now, the former explanation would be, i.e., that it's just a hallucination. That would be favored by orthodox materialist neuroscientists, right? Because there's no way that anyone, a sensible, serious neuroscientist, could entertain the idea that your brain is receiving information from some alternate reality. So that's the orthodox explanation. However, it does have its problems. As far as we are aware, the world that your brain builds is the world it evolved and learned to build. Your waking world, normal waking world, is a model of the environment that was your brain evolved and developed to construct. Your brain wasn't dropped to earth ready to construct a model of reality. It had to evolve and learn to do so using sensory information. So it's a fascinating question: is how is it possible that the brain, having evolved the function of building a model of the environment that we are born in, also knows how to build this other reality? And as you said, this is not, it's not like everybody goes to a completely different reality. Yes, the reality is very, very complex and varied, but there are qualities and content and structure within the DMT space that are seen across countless individuals. Graham Hancock here described it as highly artificial, constructed, inorganic, and in essence technological. This hyper-technological reality filled with extremely intelligent beings that are eager to communicate with you. That, to me, that's confounding. It's very difficult to explain why perturbing the brain's world-generating machinery would suddenly cause it to start constructing a bizarre alien reality filled with intelligent beings, unless it was being modulated by some kind of data source. There was some kind of information coming from somewhere else that allowed the brain to basically test its model, just as it tests its model in the normal waking life. But of course, that problem, that doesn't solve the problem, because then you have to ask, well, where's that information coming from? That's even, that's another tricky question. Where is this information that somehow your brain is able to get, that DMT is able to gain access, your brain access to in its presence? That's, yeah, it remains something of a mystery. So I can't give you a definitive answer, I'm afraid. But hopefully I'll make the problem clear. Sorry, carry on.
So somebody asked if they can get your email. You have your website also that they can reach you, right?
Yeah, that's the easiest thing to do. If you go to my website, there's a contact button, and that will send an email. I mean, the email, kind of the public email is buildingalienworlds@gmail.com. But you can do it like that, or you can go via the website. It will email me and speak through that.
It's alieninsect.org, right?
Alieninsect.net.
Oh, .net, okay.
Or buildingalienworlds.com. They both point to the same website, so either's fine.
We have a question in the chat from Oliver and Sarah saying, since the brain on psychedelics is constantly needing to update its world model, does this account for why psychedelics can induce neurogenesis?
That's a good question. The short answer is we don't know. We know that, so what you're seeing now, as I said, so psychedelics are, they're perturbing a complex system, and they're binding to all these different receptors. And I showed this neat little pathway from 5-HT2A receptor to closing of the potassium channel, as if it was kind of this simple pathway. But in fact, these pathways interact with lots of other molecules, and they actually have effects also on protein synthesis as well. So when you activate a 5-HT2A receptor, you also activate certain patterns of protein expression. And my hunch is that it's the peculiar patterns of protein expression that have been shown to be induced by psychedelics that are responsible for the growth of new neurites. So basically, you get new branches on the dendritic tree. I'm not sure. Yeah, and this is described, as neurogenesis in the form of new synaptic connections. That might be, that could also be a response to the effect that you described there. I think at the moment it's an observation. It's observed that psilocybin specifically causes neurogenesis. I think these are the sort of questions that need to be asked: is what is actually going on there? Is it a basic effect at the neuronal level, caused by something like a change in protein expression, or is it actually something that's occurring at the network level? The overall effect of psychedelics on the world model, and perhaps because the brain is needing to kind of update, it might, and columns are speaking to each other that weren't speaking to each other before. This also can stimulate growth as well. When neurons that fire together wire together. So when you start getting neurons speaking to each other that didn't before, they often form quite long-term connections as well. So psychedelics, they kind of Amanda Fielding describes it as like shaking up a snow globe. You're basically shaking up this world model, and also something that I didn't talk about, but also shaking up the model of yourself as well, and who you are and where you are in your life. And these are also models, and often they can, people who are depressed or anxious, for example, you can think of them having a non-functional or dysfunctional model of themselves. If you think that you're, a fucking waste of space and that your life is, worthless and you see no future, this is a particular pattern of thought. It's a particular part of your model of yourself and your environment. So it's not just about when I talk about models, I'm not just talking about the model of the environment, a kind of purely kind of structural thing, but I'm also talking about the model of yourself and how you see yourself interacting with your environment, and how you're looking into the past and how you're looking towards the future. That's all part of what this brain is doing. And psychedelics seem to shake all that up, which is why you get kind of ego dissolution, if you're shaking up the self model, if you like, temporarily. And hopefully, if you are suffering from something like depression or post-traumatic stress disorder or some other psychiatric condition, the idea is that you can shake up those bad models that are causing so much pain and suffering, and help to reform more functional models. And so I think that's why psychedelics are going to be so revolutionary, really, in psychiatric medicine in the very, very, very near future.
Yeah, I mean, thank you for touching on that. I mean, wait, just real quick, everybody, I sent it earlier in the chat, but I'm gonna send it again. Here's the attendance form. But anyway, yeah, I mean, this has been an awesome taste, and it's definitely leaving me with many more questions and all kinds of stuff. But seeing as we only have 1 minute left, Dr. Gallimore, if there's anything else that you want a group of Berkeley students to leave this talk with, now's the time.
Well, I mean, I would say, if you're thinking about, I mean, you're young and you're thinking about your career, and psychedelics is already exploding. Psychedelic science, and it's been chugging along, and it's been increasing over the last couple of decades, particularly. But we're now at the stage where it's exploding. Not just in terms of basic fundamental understanding of psychedelics, but also their applications. And you're going to be seeing psychedelics find their way into all aspects of medicine where the brain is involved, I think. And so if you are thinking about what to do with your life, I think the psychedelics is certainly something which you should think seriously about.